Hi, guys. So today we are here to talk about membrane transport. And specifically, we want to focus on active transport in this one. Okay, so let me get myself out of the way. Okay. So when we're talking about transport across the membrane, active transport is going to require energy. The reason it must have energy is because we're going against that concentration gradient. So think of it this way. The bell has rung, everybody else is already crammed in the hall, and you have to push yourself out of the classroom into the hall. That takes a lot more energy than walking out into the hall when nobody else is there. So typically for this, we're going to use pumps, such as the sodium potassium pump, or we may use a co-transport system. It's almost like they're holding hands, so when one goes, the other one goes too. We have exocytosis, where we're basically, it's almost like you're pooping it out of the cell. We have a vesicle, and it joins with the membrane and then opens up and releases the material out of the cell. And endocytosis, which of course we wrap the membrane around to create a vesicle, bringing into the cell. We practice that in the hallway with me standing on the stool. Okay, so when we're talking about active transport and we said it requires energy, what we're saying is it requires ATP, adenosine triphosphate. That's going to come up over and over and over just in case you don't remember it already. So this is the energy source used by cells. If I have a vehicle that uses diesel fuel, this would be my diesel fuel. It's the only thing that works. Or think of it this way. If you're going down to buy something, you can't use a euro. You have to use US money. ATP would be that US money. It's the part that the cell can use. We have other energy molecules, but we have to turn them into ATP to be used by the cells. So it's used in active transport. That's the energy that is required. And the way it works is, you notice we have our adenosine triphosphate. Here's 3 phosphates. And so what has happened is that we can transfer that terminal phosphate group to the transport protein. And with that phosphate group is where the energy is stored, kind of in the bond there. And so when we transfer it, it changes the shape of the transport protein so it actually can move the substance more easily. And so we refer to this as a conformational change because we're actually changing the physical shape. So first of all, we focus with pumps. Pumps are often used to maintain a certain membrane potential, especially when we're talking about muscles or nerve impulses. The way these are controlled and the way these function have to do with a certain variation between a positive and a negative charge on each side of the membrane. And so sometimes we have to pump certain charged particles to one side of the membrane over the other so that we can create this membrane potential, this variation. So it's an unequal concentration of ions. Often sodium or potassium those are the most common ions that you're going to find that we use for these unequal concentrations. So we end up with this electrical charge. Like I said, we're having more ions on one side than the other, so we're going to have more plus or more minus. Cytoplasm is relatively negative in comparison to the extracellular fluid. So that's what's going to help us move it. So we can have an electrogenic pump. In this, the proteins actually generate voltage across the membranes, and we can use that later kind of as an energy source for cellular processes. So our sodium potassium pump, in animal cells, it actually regulates the concentrations of sodium and potassium across the membrane. We get 3 sodiums pumped out of the cell and 2 potassiums will get pumped into the cell. So we'll send out 3 pluses and bring in 2 pluses. So every time we use that sodium potassium pump, we get a net +1 charge to that extracellular fluid because we put 3 pluses out and only brought 2 in. So we ended up with an extra plus outside. Make sense? Hopefully. Okay, then we could have a proton pump. So plants, fungi, and bacteria will typically use proton pumps, and they will actually pump hydrogen out of the cell. Okay. So here's our picture of our sodium potassium pump. So we've got lots of potassium inside the cell compared to sodium outside the cell. And so animal cells have a relatively low concentration of sodium inside compared to outside. So what happens is our ATP phosphorylates, puts the energy that causes the conformational change that leads to move the sodiums out. And notice when it does so, the new shape now prefers the potassium, so it's going to pick up its 2 potassiums. And that K+, when it binds, it basically causes that phosphate group that we added on from ATP, it causes it to be released, so it lets go, causing the cell to go back to its original shape, bringing the potassiums into the cell. So the phosphate group is gone, the protein came back to the original shape, and it moved those potassiums in when it did that. And then we have what we call co-transport. So the idea is that we're going to couple a favorable movement, so something that we want to easily move across the membrane, with what we consider an unfavorable movement, some other source. So favorable would be one of those downhill diffusions, so we're going with the concentration gradient. Unfavorable would be the opposite, we're going against the concentration gradient. So for example, plants will use co-transport for sugar and amino acids. So the idea is that sucrose with a hydrogen ion are co-transported. So the sucrose can go into the plant cell against the concentration gradient if it's coupled with that hydrogen ion going down the gradient. So think of it as the hydrogen ion is allowed to come in and it's holding the hand of the sucrose, and because they're holding hands and holding on tight, when one goes, the other one is allowed to go with it. And so here's our sucrose. They're holding on to the little hydrogen, and because the hydrogen's allowed to go across, it brings the sugar with it. Okay, and so then we have really large molecules, and the big molecules, typically we use exo and endocytosis. So exocytosis is where we're secreting molecules using vesicles out through the plasma membrane. So they're going released out into the extracellular fluid. For example, nerve cells release neurotransmitters like dopamine. Then we have endocytosis to take up molecules from vesicles that fuse to that plasma membrane. So it's going to be the opposite. So for example, phagocytosis and pinocytosis. And remember, phagocytosis is going to be something that we're taking in something large. Pinocytosis is taking in something small, almost like it's drinking. And then we have one that's receptor mediated. Okay, so phagocytosis, the cell's engulfing a particle that's going to get taken in. You'll notice it's been taken in as a vesicle, and then it will be digested by lysosomes so we can use the parts. Okay, so we're going to package it and fuse it in with the lysosome, and then we can digest it and use the materials. For pinocytosis, we have these much smaller particles that are kind of dissolved in there. That's why I said it's almost like it's drinking. But the process is the same, where we're taking in those materials into a vesicle. And then that vesicle will come in and it will transport and we'll go through the same process. And then we have the receptor mediated endocytosis. So it's the same process in terms of taking in the materials, but it only works if the material has properly attached to the receptor, signaling that uptake. So it allows the cell to take large quantities of that substance because we attach to those receptors saying, okay, let's do this. And so notice we've attached the little triangles to the receptors and we've picked up the receptors and the other material into our little vesicle. Okay, ooh, we got a practice free response question. You want to determine the effect of pH on the uptake of sucrose in tomato plant cells. You expose the cells to a sucrose solution at varying pH levels and record the data, which is seen in this chart here. So at pH 4, you can see that's our green line. At pH 6 is our red line, and pH 8 is our blue line. So the first thing they ask you to do is identify the optimal pH for sucrose uptake in tomato plant cells. So which one does it take up the most sucrose? And then it asks you to describe why the pH identified in part A would lead to an increase in sucrose uptake. Okay. And then another practice-free response. When a pathogen, such as bacteria, invades the human body, the immune system is alerted. Once alerted, white blood cells, such as macrophages, locate the pathogen and engulf it through phagocytosis. So A, describe the process of phagocytosis. So here you would explain to me in a little paragraph how phagocytosis happens. Okay. And then B, identify the most likely component of the plasma membrane that the immune system uses to recognize the pathogen. So think about what would it happen with the plasma membrane so it knows, oh, this is what I need to take in, because we don't want to just take in or phagosize the wrong particles. So what do you think we might do? What might we use to make this possible? Okay, so hopefully that helps and you got a little something from that. Bye.